What's a prokaryotic cell? A. A, ty- a type of cell found in plants and animals containing a membrane-bound nucleus and other membrane-bound organelles. B. A type of cell found only in plants, characterized by a ligid, ligid cell wall and chloroplast for photosynthesis. C. A type of cell found only in animals, categorized by a lack of a nucleus and membrane-bound organelles. D. A type of cell found in bacteria and archaea, la- lacking a membrane-bound nucleus but containing genetic material.
Thank you very much, Sora. So let's have a few more seconds. One more person? Perfect. Okay, so here are the results. Sora, what have you why have you chosen D?
I chose D because prokaryotic cells is bacteria and archaea. So A, B, C is not written about bacteria and archaeas. So I choose D.
Mm-hmm. Very well. Perfect. So all of you went for correct option. That's the great then. Um, type of cell found only in animals. Okay, so this one, as you said, was incorrect regarding C. What's wrong with B, guys? With B, what's wrong with B?
Chloroplast for photosynthesis is not correct.
And also found only in plants. Okay, perfect. As you said, Misato, great. Chloroplasts for photosynthesis are typical for plant cells, which are eukaryotic cells. And also, the prokaryotic cells are not to be found only in plants. And the A1, the A option,
Found in plants and animals.
perfect. Found in plants and animals because containing a membrane-bound nucleus and other membrane-bound organelles. These would apply for eukaryotic cell. As we remember from our first session, we were comparing those two, and as you know already, there are no membrane-bound organelles as we have here. Endoplasmic reticulum, Golgi apparatus, mitochondria, and so on. And also, nucleus is encapsulated in the membrane. Whereas in the prokaryotic cell, we have just a nucleoid, which is circular DNA floating in the cytoplasm, and there is no membrane separating the DNA from cytoplasm space. So very well, all of you handled the question great. Okay, let's move then. Um, now, please, can you read?
What is exocytosis? Exocytosis A. The process of bringing substances into the cell through the cell membrane. B. The process of breaking down large molecules into smaller ones within the cell. C. The process of expelling substances from the cell through the cell membrane. D. The processes of producing energy in the cell through cellular respiration.
Thank you very much. Now, let's have a few more seconds. Still three more people to. Okay, two, one. One more. Okay. So, guys, here are the results. Now, what have you chosen and why?
I chose C because A is into the cell through the cell membrane is not correct, and B is breaking down large molecule into smaller ones is not correct, and D is producing energy is not correct, so I chose C.
Very nice. Okay, process of expelling substances from the cell through the cell membrane. Exactly. Exocytosis is a process when uh, uh, involves the release. Let me show you the picture of the content of the vesicles. Okay, we mentioned examples such as pancreatic cells, for example, and insulin as a substance. Um, However, we also mentioned marginally that it can be part for neurotransmission, or neuronal cells are able to communicate via exocytosis process. If you remember, I draw something like this. This is one part of a neuron. This is another part of neuron. Okay, and here between them we have like a gap. Okay, there is a gap. We call it synapse. And in this synapse, we release neurotransmitters. Okay, neurotransmitters. From neuron 1 to neuron 2, neuron 1 synthesize, and then release neurotransmitter via exocytosis in order to communicate and transmit the signal onto the second neuron. Okay, and this is only via exocytosis, so active way of transport when we use an energy for releasing those neurotransmitters onto another neuron. So very good. um, the process of breaking down large molecules into smaller ones within the cell. Do you remember, guys? We mentioned basically two groups of metabolism. All of the reactions can be grouped into one or second. Do you remember the names of it? First one is when we are creating or more complex substances are made, and the second one is when the substances or the complex large molecules are break down uh, into smaller one into release the energy. Do you remember? Two types or two metabolism, basically two groups. Is there anyone who remember? The first one was anabolic. The second one was catabolic. I'm writing it now here. Anabolic metabolism, catabolic and metabolism. The process of breaking down large molecules into smaller ones within the cell. What do you think? Is it anabolic or catabolic? Do you remember anyone? During anabolic processes, we create or we are made we made make We are making a more complex and larger molecules from smaller ones. Anabolic, anabolic are prevailing during childhood, or they are typical for children when they are growing. Children are growing, so they are creating um, the stronger bones, more muscles. They are growing. Their neuronal web is is growing as well. Organs are growing. Everything is getting trying to be bigger and more mature. So anabolic metabolism is prevailing. On the other hand, catabolic metabolism is the way when large molecules are breaking down into smaller ones into release the energy. Catabolic metabolisms are prevailing in older people. Okay, perfect. So, what would be the name for B? What kind of metabolism?
Catabolic metabolism.
Perfect. Mhm. What is the process described in the first option, in the A? The process of bringing substances into the cell through the cell membrane.
Endocytosis.
Perfect, perfect, perfect. Endocytosis. So basically the total opposite of exocytosis. Very well. Okay, let's move further. What's going on? Okay. Yuki, please, can you read?
Yes, what is
Yuki, a little
transcription
bit
in
more louder or come closer to the microphone because I can't hear you.
okay. What is transcription in the context of molecular biology?
Yuki, I think there is a problem on your side. Maybe microphone is not working properly because I cannot hear you. Maybe I don't know if others can. Guys, can you hear Yuki? No.
Yes.
Mm. Okay, Yuki. I think maybe try to refresh and the other from our colleagues will read. Misato, please, can you read?
Yes. What is transcription in the context of molecular biology? A. The process of making a copy of DNA using RNA polymerase. B. The process of synthesizing proteins from messenger RNA template. C. The process of converting RNA into DNA. D. The process of translating messenger RNA into proteins.
Thank you very much, Misato. Okay, here are the results. So let's discuss them. Misato, what have you chosen and why?
I chose A because I think mm, B, C, and D is translation part.
Okay, so you went for A, right? So let's propose A is correct. The process of making a copy of DNA using RNA polymerase. You are right. Transcription. Yes. Here we are. If you zoom in a little bit, it will be obvious that here we have DNA and here we have RNA polymerase, which is enzyme responsible for synthesizing mRNA based on DNA. Okay, all of it happens in the cytoplasm. If we have already synthesized messenger RNA, it goes through the pores in the nucleus membrane and travels to the cytoplasm where it bonds itself to ribosomes in order to continue with the process we call translation. As Misato, as you mentioned, the B would be process of synthesizing proteins from mRNA templates would be translation. Very well. The what about D? Is it also translation? Guys,
I think this is also written about translation.
perfect. Yes, it is. Yes, I was just checking. Um, if you are aware, great, great, perfect. However, C is not the process of translation. C is something we are not going to deal with shortly or even during this course. Nevertheless, something like the process of converting RNA into DNA exists, and we call it reverse transcription. Okay, so the transcription in the opposite direction. However, only few organisms are able to do it, and one of them is HIV virus, basically. Okay. Maybe you already heard about HIV virus before. HIV virus attaches your cells and it has an RNA inside of it, and it can then transcribe and produce DNA which will get into your DNA, and this is the way how this virus infects our bodies or the bodies of humans via reverse transcription. Okay, so it's very clinical thing from a higher level of medicine studies. Nevertheless, this is not translation. Okay, this is not translation, something different. Okay, let's go then further. Satsuki, please, can you read?
What is translation in the context of molecular biology? A. The process of making a copy of DNA using RNA polymerase. B. The process of synthesizing proteins from mRNA templates. C. The process of converting RNA into DNA. D. The process of transcribing DNA into mRNA.
Perfect, thank you very much, Satsuki. Let's have a few more seconds. Okay, here are the results. Let's discuss them. Satsuki, what have you chosen and why?
I chose B because A and D is transcription and C is reverse transcription.
Okay, so which, sorry, I lost myself. What did you went for? Which option? You
B.
B. Okay, so you went for B. Sorry. The process of synthesizing proteins for mRNA templates. Okay, so Satsuki suggested the B. Exactly. Okay, process of synthesizing proteins from mRNA. The A, the process of making a copy of DNA using RNA polymerase refers to. Satsuki, can you just repeat what you said?
Sorry, one more time, please.
The A, you already mentioned that. The process of making a copy of DNA using RNA polymerase. This process describes what?
Transcription.
Transcription. Great. Mhm. Making a copy, yeah. The process of converting RNA into DNA is what?
Reverse transcription.
Reverse transcription. Exactly, perfect. We just discussed that, but it's not relevant for you, but it's like additional information. Great. And the process of transcribing DNA into mRNA? Anyone?
Transcription.
Again, perfect. Great. Great, great, great. Okay. Where does the translation takes place in the cell? Where?
Cytoplasm.
Okay, cytoplasm on ribosomes. Transcription?
Inside of the nucleus.
Very well. Thank you very much. Okay. Now, Yuiko, please, can you read?
What is replication in the context of molecular biology? A. The process of making a copy of DNA using RNA polymerase. B. The process of synthesizing proteins from mRNA templates. C. The process of converting RNA into DNA. D. The process of making a copy of DNA during cell division.
Mhm. Perfect. Thank you very much. Okay, here are the results. Let's discuss. Yuiko, what have you chosen and why?
I choose D.
Okay, and
Uh,
why?
uh, I think A is transcription and C is like reverse transcription, and I think the B is not incorrect. Incorrect.
Hmm. Okay, so you went for D, Yuiko. Perfect. Okay, let's propose that's right. Um, as you said, C is the process of converting RNA into DNA is reverse transcription. The process of synthesizing proteins from mRNA templates. How do we call this process?
Reverse transcription.
It's a C. Reverse transcription is the process of converting RNA into DNA. That's right. B is, however, different process when we are synthesizing proteins from messenger RNA templates. Do you know?
Translation.
Translation. Okay. However, why A is incorrect? The process of making copy of DNA. It's almost the same as the D one. What's wrong then?
Using RNA polymerase is not correct.
Perfect. Because using RNA polymerase is, you know already that it has something to do with RNA. Okay, here please zoom in a little bit. We are able to see that RNA polymerase it's creating a polymer of RNA. Okay, perfect. So it's something different of replication because, as we remember from replication, we have a DNA polymerase which is responsible for replication as an enzyme. Okay, very similar name, but totally different process. Okay, DNA polymerase is responsible for replication, whereas during transcription, RNA polymerase plays a crucial role. Okay, so you went for this very, very correctly and you found it. Okay. Um, uh, Last activity. Guys, let's have 1 minute and your task is to connect those nitrogenous bases with correct nucleic acid. Let's start. 1 minute. Individually, okay? Okay. So time's up, more or less. So, guys, what about adenine?
In case of DNA, adenine to thymine
Oh,
is the
yeah,
set.
that's right. However, are we able to find adenine in both in DNA and RNA? That's the
Oh
question.
yes. Oh yes, yes, yes.
Perfect. So please, Sora, can you link adenine with the red color to DNA and with the blue color to RNA?
Okay. How to change the color?
In
Ah,
the
okay, 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 okay,
okay,
okay, okay.
perfect. Thank you very much. What about guanine?
DNA and RNA has guanine.
Perfect. Misato, please, can you do the same um, with the red to DNA and blue to RNA?
Yes. Ah, I'm sorry, I mistake.
It's okay, it's okay. Now it's correct. So we see.
Ah,
What about thymine?
Only DNA.
Perfect. Yuiko, please, can you link it with the red one? Thank you. Uracil. What about uracil?
Only RNA.
Perfect. Please link it, Sora, with the blue one. And last one, cytosine.
DNA and RNA.
Perfect, Satsuki. Thank you very much. Please link it. Perfect. So, as we see from our chart now, guys, adenine, guanine, thymine, and cytosine belong to DNA, and adenine, guanine, uracil, and cytosine belongs to RNA. Here, yesterday we saw the picture where we this also divided them into two groups, purines and pyrimidines. Okay, pyrimidines and purines, they are named only based on their chemical structure. Okay, you will study that more in depth in chemistry sessions. Nevertheless, do not forget, always purines are complementary with pyrimidines. Okay, one pyrimidine with another purine and vice versa. How to remember that? Okay, we mentioned this nice helper or simply held simple helper. The pointed letters are complementary and rounded letters are complementary. So guanine as a purine is complementary with cytosine with as a pyrimidine. And the same applies for adenine is complementary with thymine. In the RNA, we do not have thymine, we have uracil instead. So the letters are not now that doesn't apply to that rule. This rule applies only, or this helper applies only for DNA. But still, what applies is that one pyrimidine is complementary with one purine. Okay, so do not forget this because this is essential for admission questions. Okay, this complementarity um, and the nitrogenous basis to know them. Okay, not formulas. You don't have to know formulas, but you definitely have to know which one belongs to which nucleic acid. Okay, guys. We are okay. We should now continue with the core. However, we still have 5 minutes. So I suggest, or would you like to have break now for 5 minutes and then we will start with the all core, or would you like to continue for 5 more minutes? Guys, it's up to you. Yes, I want break now. No, I want break in 5 minutes. Okay, so you don't want have break. So we will continue now. Okay, let's continue. Um, core cell cycle. Yesterday we started to describe everything or try to describe the chromosome. Okay, um, here we are able to see the cell cycle. First of all, we mentioned that the cell cycle is exclusively associated with the cell division. That means that when the cell is undergoing the interphase or mitosis, during interphase is preparing herself or itself for upcoming cell division. Nevertheless, there are cells which can stay and exit, which can stay in G0 and exit the cell cycle. What does it mean? Basically, the cells or a lot of cells in adult organisms are in G0 phase. So they are not preparing, growing, or undergoing any processes in order to get to cell division. And basically, there are two groups of cells. One cell groups can or remain in the G0, but they can re-enter the cell cycle. Those cells can be, for example, skin cells in our skin or mucosal cells, okay, on our mucoses. Those cells remain in G0, but if there is some stimuli or triggering factor, they can re-enter. The other group of cells cannot re-enter the cell cycle at all. Can you guess maybe or provide just one example of cells which would remain till our death in the G0? Any ideas? Which cells cannot get into cell cycle, cannot divide anymore, and will remain in G0? Any guesses? You can try. Whatever comes to your mind, we can try. Okay, I can help you with. We mentioned the neurons already today, and neurons are as an example of those cells. Neurons are the cells of neuronal system cannot divide themselves anymore. The same applies also for our hearts. So cardiac muscle cells cannot divide anymore. Or we already discussed many times erythrocytes. Erythrocytes also remain for the majority of their life in G0. They cannot divide themselves anymore. If we want more erythrocytes, we need to undergo a transfusion, or our bone marrow needs to produce new erythrocytes, but erythrocytes themselves cannot divide. That applies also for neurons, that applies also for cardiac muscle cells. Okay, so that's basically how to understand G0 phase. During G1, yes, and there is this thing regarding diploid and haploid. Okay, it's 55. Guys, do you want to have a break now, or you want to continue without break? Yes, I want break. No, I don't want break. Okay, so you want 5 minutes break. Let's have 5 minutes break. Okay, now see you in 5 minutes. Okay, guys, are you back? Okay, so let's discuss a cell cycle. Yesterday, you started to work on the phases. Can I ask the group responsible for G1 phase to present your findings about the G1?
Yes, in G1 phase, cell growth and preparation for DNA replication occur, and in G1 phase, there is a checkpoint that checks whether there are substances that promote division, whether the substances necessary for division are not accumulated, and whether there is no damage to DNA. That's
Mhm.
all.
Perfect. Checkpoint. Okay. This is very important to mention. Um, checkpoint and growth. Those two things you should remember. Growth during G1, and exactly as you said, now G1 is a post mitotic phase. Basically, it's after mitosis. And again, it prepares for another cell division. Um, the checkpoint you mentioned is very important because before the S phase, when the DNA replication is happening, the cell is checking herself whether everything is okay, whether everything is right, if there is no error in the DNA or in the genetic material. So this is the first checkpoint. However, if something goes wrong and the cell detect some error or issue, there are two possibilities what can happen. The first one, the first one is that the cell will stop the cell cycle and will not continue in a cell cycle, or if the error is more serious, the cell undergoes apoptosis. Do you remember what apoptosis is? We already mentioned that. Apoptosis.
Cell is killing itself.
Perfect, yes. So it's programmed cell death. So if the cell during a checkpoint part of G1 phase finds out that there is a problem, the cell can kill herself, suicide herself basically. Okay, perfect. During G1 phase, is important to mention that the cell has functional organelles, it's ready to perform its functions, and it has a diploid set of DNA. Guys, I would like to know whether you are familiar with the haploid or diploid set of DNA. 1N haploid, 2N diploid. Yes, no? Okay. Okay, so most of you went for yes. So please explain to us what is haploid, guys. What is it to understand that? Anyone?
Haploid is difference of amount of DNA during cell cycle.
Um, haploid set of chromosomes is different regarding the number, definitely. However, it is also connected to cell cycle, but it's a little bit different. Um, the meaning. Basically, the haploid means that the set of chromosomes is half, and there are specific cells where we can find them in the body. Do you know? Guys, would you like to maybe see a video, a very short video to break that down, these very abstract terms, differences between haploid and diploid? Okay, one more person. Okay, so most of you went for yes. So let me very shortly provide you with an explanation of these abstract terms to break down uh, the differences between haploid and diploid. 1 second, yes. So I need to desktop share, start screen share, share.
In this video, we'll discuss haploid versus diploid cells. Haploid and diploid are terms that describe the number of sets of chromosomes in a cell. Haploid means a cell has only one set of chromosomes, and diploid means a cell contains two sets of chromosomes. In your body, sex cells, called gametes, have a haploid number of chromosomes, represented by the symbol N. In humans, every gamete has one set of 23 chromosomes, so the haploid, or N number in humans, is 23. This is important since the union of gametes during fertilization creates a diploid cell called a zygote, with two sets of chromosomes, for a total of 46. At fertilization, the chromosomes from each parent match up to become the new pairs of chromosomes in a zygote. Each pair contains one chromosome from the father and a corresponding chromosome from the mother. These pairs are called homologous chromosomes. Homologous chromosomes are similar in shape and size, along with the same types of genes in the same locations. A diploid zygote will go through cell division many times to produce all the cells in the body of a fully developed baby. All body cells, except gametes, are referred to as somatic cells. In humans, somatic cells are always diploid, written as 2N, which means they have two sets of 23 chromosomes for a total of 46 chromosomes. Other organisms have somatic cells with different diploid numbers of chromosomes. But the gametes in these organisms are haploid, meaning they always have half the diploid number of chromosomes. So how does cell division affect the number of chromosomes in daughter cells? Well, somatic cells only reproduce by mitosis, a type of cell division that results in two genetically identical diploid daughter cells. In contrast, meiosis is a type of cell division that only produces gametes. In meiosis, a diploid cell undergoes two cell divisions to produce four genetically different haploid gametes. We'll cover the details of meiosis in another video. In summary, diploid cells have two complete sets of chromosomes, one set from each parent. Diploid cells have twice the number of chromosomes as haploid cells. The two sets consist of pairs of homologous chromosomes. The diploid chromosome number is written as 2N. All somatic cells Whether they're skin cells, muscle cells, or leaf cells in a plant, are diploid. Diploid cells reproduce only by mitosis. And gametes are never diploid. In contrast, gamete cells, which are always haploid, have only one set of chromosomes, which is half the diploid number. Since there's only one set of chromosomes, there are no homologous pairs. The haploid chromosome number is written as N. All gametes are haploid. And haploid gametes form from diploid cells through meiosis, never through mitosis.
Okay, guys, so hopefully now it's clear, understandable, the difference between haploid and diploid. So if we start G1 phase, the cell after mitosis is 2N, so it has 46 chromosomes and it's somatic cell, okay? And what's going on? What's going on? We already said that during G1 phase, there is a checkpoint and there is a growth of a cell preparing for S phase. What is upcoming phase? We are going to there shortly. Okay. We mentioned that if there is some problem or error detected during checkpoint, the cell cycle can be stopped, or even in the other way, the cell can undergo apoptosis or it can suicide herself. Okay, that brings us to the S phase. Please, the S phase group, who was it yesterday? Okay, Sora, you are raising hands, so go on.
Okay. S phase is synthesis with DNA.
Okay,
That's all. That's all.
that's all?
Yeah.
Um, okay,
Not
so,
enough.
um, sorry?
Is it not enough?
Like more explanation would be beneficial, but we will cover that together. Yes, you are right. S phase is basically a synthesis of DNA. What does it mean? You know, there is much more to add basically. That's why you have more time in the breakouts to search more to be more clear. So, S phase. Let me show you the picture. During the S phase, the replication of DNA is happening. This is the thing we discussed uh, lately, and we were starting to discussing also today. So replication, semi-conservative method. How do we synthesize new DNA? What does it mean, the semi-conservative method? Do you remember, guys? Or process? Semi-conservative. What does it refers to? As we see, there are at the beginning one double-stranded DNA molecule, and at the end we have two double-stranded DNA molecules. However, they are in different color. What does it mean? That this is the old molecule, and it's in a blue color, and the newly synthesized here is in a purple. And semi-conservative means that it contains one old strand and one newly synthesized, the purple one. Okay, so one is old, one is new. They are complementary, and we have now doubled the amount of DNA basically. Even though they contain the same genetical information, they are doubled in order to have enough genetic material for mitosis. Okay, so what does it mean basically, the S phase? So let me get back to the. So what how does it look like if we are checking the S phase on this picture regarding the replication? As you know, we describe chromosomes when they are in this form. However, most of the time, or most of the time for a cell, chromosomes doesn't look like this. This is a appearance, how they look like during metaphase of mitosis. There are more phases, 4 phases. We will get there today. And during metaphase of mitosis, the chromosomes look like this. You remember we discussed yesterday. We have one centromere, longer arm and shorter arm. And if we have on one chromosome also another centromere, those, chromatids, my bad, chromatids. We have only one centromere here in the middle, and chromatids. My bad, I apologize. My bad. One chromatid on this side and the second chromatid on the left side. Both, this one and this one, have the same DNA information. So the information on this spot is the similar to the information on this spot. Why is it important? Because usually our chromosomes looks like this. One from mother and another from a father. This is one chromosome and it is a homologous pair of a chromosome. Okay, so this is one pair of chromosomes. So one pair, one chromosome, another chromosome. We have one pair, oh, one pair, and totally in our cells there are 23 pairs, okay, of chromosomes. So that makes 46 chromosomes in total. This is one chromosome and it has one chromatid. Right? So far, guys? After replication, we still have one chromosome, but two chromatids on this picture. And it's very important now because after replication, we still have 46 chromosomes, 23 pairs, but 92 chromatids because our amount of DNA has been doubled. It was replicated. Before S phase, before replication, we have 46 chromosomes and 46 chromatids as well. Okay, as you see on this picture before replication, one more time, we have a longer arm of chromatid, shorter arm of chromatid, and together this is the all chromosome basically. Okay, just one chromosome. After replication, we have two sister chromatids on one chromosome, and together we still have 23 pairs of them. We still have 46 chromosomes, but we have doubled the amount of chromatids. Before replication, it was one chromosome with one chromatid. After, we have one chromosome with two chromatids. Understandable so far, guys? Yes? Okay, perfect. Because this is very important and also very often confusing for many people. Okay? So, that was the S phase, basically. Um, yes, I think that's all we need to know so far, we need to cover. And when the DNA is replicated, it takes some time, yes, and that brings us to the G2 phase. Guys, who was responsible for G2 phase? Yuiko, you are raising hand. Go on.
Yes, okay. G2 is period of preparation of cell division. Protein synthesis in this process. That's all.
Okay, yes. There is also but one very important thing to mention. You are absolutely right. So during the G2, the cell itself is still preparing for mitosis. So as you mentioned, like synthesis of proteins, also multiplying the amount of organelles in order to have the same amount and the sufficient amount of organelles in both the other cells. And what is necessary to mention additionally, there is another checkpoint. Okay, here we have another checkpoint where cell checks her. Do you understand, guys, the verb check? What does it mean? Yes? Okay. So basically the cell check herself if everything is all right, if everything is correct, even after replication, if there is no error in the genetic material, if the cell is basically working properly. However, if there is an error detected, if there is a problem, the cell will undergo apoptosis. Okay, so cell will suicide itself. Okay, so we have two checkpoints during interphase in G1 and in G2. In the middle or between those two phases, we have a S phase or synthesis phase where the DNA is replicated. Okay, so this is the things or all of those facts you should be aware and you should know um, for the test for the admissions. What is going on in which phase, basically? Okay, and that was the interface. Um, uh, yeah, let's do a little check quiz. Utah, please, can you read?
Which of the which of the following cell cycle description is correct? A chromosomes chromosomes
Chromosomes.
uh, chromosomes are replicated during the G1 phase. Interphase is when the cell spends most of its time performing its cell functions. The S phase consists of prophase, metaphase, anaphase, and telophase.
Thank you very much, Utah. Two more people. Okay, so let's discuss. Here are the results. Yuta, what have you chosen and why?
Sorry, I missed up the.
Mhm.
I chose A, but I think B is correct.
Okay, and what's wrong with A? What do you think?
Not G1 phase.
And which one?
I think G2.
G2, there is a checkpoint whether chromosomes are replicated correctly. However, the other phase is right when the replication takes place. Let me show you one more time the picture. We also know that during G1 there is a growth and preparation for mitosis, and during S phase DNA is synthesized or replicated. So in the red one. So chromosomes would be replicated during the S phase. Okay, so if the statement would be written chromosomes are replicated during the S phase, that would be correct. Okay, most of uh, most of you went for B. That's great. And the S phase consists of prophase, metaphase, anaphase, and telophase. That doesn't This is not correct regarding S phase. This is correct regarding mitosis, where we are heading now. Okay, mitosis. Okay, guys. I think that's overall everything we have to know regarding interface. Now, let's move for the mitosis. Um, we have options how to start with mitosis part. Um, we can start, guys, that I will play a video to understand this abstract or to visualize this abstract process initially, or I will start to explaining those phases to you and then we will watch a video. So what would you like to do? First, A is video first, B is verbally first. Okay? Okay, so most of you went for a video first. So let's do the video now to understand what or to see the mitosis, to understand mitosis. Let me start screen share. Yes, share big screen now.
In this lesson, we'll be exploring the M phase of the cell cycle, including mitosis and cytokinesis. Let's do a quick review of the cell cycle to see where they fit in. The G1, S, and G2 phases make up interphase. And the M phase represents cell division. Cell division includes division of the nucleus, called mitosis, and division of the cytoplasm, called cytokinesis. Mitosis is further broken down into 4 phases: prophase, metaphase, anaphase, and telophase. Prophase is the longest phase of mitosis. Prophase is when chromatin begins to condense into the shape of chromosomes, and the nucleolus disappears. The previously replicated DNA coils tightly into sister chromatids. For the first time, you see individual chromosomes. In the center of each chromosome, a centromere attaches the sister chromatids together. Meanwhile, in the cytoplasm, microtubules, known as spindle fibers, begin to fan out from two sets of paired structures, called centrioles. The spindle fibers elongate as the centrioles begin moving to opposite sides or poles of the cell. While this is happening, the nuclear membrane surrounding the nucleus disappears. Now that chromosomes are no longer separated from the cytoplasm, the opposite ends of the spindle fibers can attach to the centromeres. Next, the cell enters metaphase. The centrioles complete their movement to the poles of the cell, while the spindle fibers line up the chromosomes along the equator of the cell. The end-to-end alignment of chromosomes results in a sister chromatid on either side of the equator. Anaphase follows metaphase. During anaphase, spindle fibers separate the sister chromatids at their centromere. Once separated from each other, each chromatid is called a chromosome. The single-stranded chromosomes form a V shape as the spindle fibers shorten and drag them through the gel-like cytoplasm. The chromosomes move to opposite poles of the cell toward their centrioles. It's common to confuse centrioles with centromeres, which connect chromatids. Remember, centrioles are at the poles. Telophase is the final stage of mitosis. In telophase, a nuclear membrane reforms around each set of chromosomes. Then, the chromosomes spread out into chromatin, and the nucleolus becomes visible once again. Mitosis, the division of the nucleus, is now complete. The final step of the M phase is cytokinesis, the division of the cytoplasm. In animal cells, cytokinesis occurs through the inward movement of the cell membrane. This progressively pinches the cytoplasm until two identical daughter cells form. In contrast, plant cells can't pinch in two because they have a rigid cell wall surrounding their cell membrane. Instead, cell wall material assembles along the equator, forming a structure called the cell plate. The cell plate grows until it joins with the existing cell membrane, separating the two halves of the cell into daughter cells. Over time, new cell walls form between the two daughter cells. Here are the key points to remember. The M phase is the fourth and final phase of the cell cycle. During the M phase, cell division occurs through two processes: mitosis, when the nucleus divides, and cytokinesis, when the cytoplasm divides. Mitosis has four phases. During prophase, chromatin condenses into chromosomes, spindle fibers form, and the nucleolus and nuclear membrane disappear. During metaphase, spindle fibers align the chromosomes along the cell equator. In anaphase, the spindle fibers separate sister chromatids into two separate groups of chromosomes, pulling them toward the poles. And in telophase, the nucleolus and nuclear membrane reform. The chromosomes disperse into chromatin. Cytokinesis is division of the cytoplasm. The M phase is complete after cytokinesis occurs. The M phase of the cell cycle always results in two daughter cells. Both of these daughter cells are identical to each other and identical to the original cell that underwent mitosis.
Okay, guys, so that was it. So it was introduction to mitosis. Okay, guys, now check whether you were aware of what we have seen. Short group work, okay? Very short group work. We will have 4 minutes only to cover your part. And let me tell you who is going to do what. Each of you will different phase to cover. So we have four phases basically. We will not do phase as it is on the picture, even though sometimes on some university resources, um, some authors or some professors can require that even phase is like individual phase. However, it is nothing else than the continuation from prophase to metaphase. Yes. Now, Misato Mona, you will do prophase. Now, Satsuki, you will do metaphase. Anaphase will be covered by Sora and Yuiko, and telophase, Yuki and Yuta. Okay, one more time. Prophase Misato Mona, now Satsuki. Metaphase, anaphase Sora Yuiko, uh, Yuki and Yuta, uh, telophase. Let's start. You have 4 minutes just to search and to recap what we have seen. Okay, guys. So let's start with the first group, prophase. What have you found out, Misato and Mona? Ladies?
The,
Sorry, yes, go on, Mona.
the process is the first phase of mitosis, the process that separates genetic material in the nucleus of a parent cell into two daughter cells.
Mm-hmm.
And uh, in the prophase, nuclear envelope and nucleus is disappeared, and the chromosome become thicker and shorter.
Mm-hmm. Perfect. Exactly. So, as you said, during the prophase, as it is the first phase of mitosis, the chromosomes are condensated and it creation of mitotic spindle. Okay, so as you said, in prophase, the nuclear membrane is disintegrated, so chromosomes are now within the reach of the spindle fibers. Okay, let me remind you where those spindle fibers are attached. Those spindle fibers are attached on our centromeres. Okay, so here the spindle fibers are attached. There can be confusion between centromere, centriole, and centrosome. Okay, guys, there are three different things referring to. Centromere is the place where the spindle fiber attached, okay, on the chromosome. However, centrioles are um, the or they contain of 9 triplets of microtubules, as we are able to see them here. Maybe I will show a different picture with what will be maybe more useful in this case. Yes, this one would be nice. So let me share my screen. Okay, here we are able to see microtubule triplet. Okay, this one thing is centriole. However, together, two centrioles make up a centrosome, what is already an organelle. Okay, and this organelle, the centrosome, contributes to the cell division. Okay, so one is centriole, two centrioles together make up the centrosome, and centromere is the spot on chromosome or the place where the where the spindle fiber attaches. Okay, what else to prophase? Yes, and as you mentioned, exactly, Misato, the chromosomes are more thick and shortened, so they are condensated. Yes, I think we can now continue with the metaphase. Metaphase. Who covered metaphase, guys?
Um, Metaphase is the spider threads attached to the chromosomes and line the chromosomes up on the equator.
Mm-hmm.
That's all.
Perfect. So equator. Here we have an equator on this picture. So it's like equatorial plane on the planet Earth. We have also equator here within the cell because it was named exactly after equatorial plane um, because it's similar. Okay, chromosomes are in the equatorial plane. Uh, what is mentioned to know, they are in maximally condensed and they are aligned in the equatorial plane. And now in this phase, in the metaphase, they are visible under the microscope. Okay, so in this phase, they were firstly recognized under the microscope. That's why they were also described on their structure here. So they have two chromatids. One chromosome have two chromatids. That's why I also learning it like this, because they were visible for the first time during metaphase under microscope. Okay. What about anaphase? Go on, Yuito.
Okay. Anaphase is chromosomes break out centromeres and sister chromatids move to opposite ends of the cell.
Perfect. That's very good. Exactly. So, separation of the sister chromatids. One chromosome, we have like X-shaped chromosome, or it would be better like, no, yeah, X-shaped chromosome like this, and it's pulled apart, so the sister chromatids goes to different poles of the cell in order to divide. Okay, one chromosome, one chromatid has the same DNA as the other one, and now the spindle fibers shorten themselves, so they are torn apart, pull apart in different poles. So that's what's going on in anaphase. Um, yes. What about telophase? What's going on in telophase?
Yes, telophase is chromosomes unravel into filaments, and
Yuki,
the new yes.
Yuki, can you maybe move a little bit closer to the microphone?
Okay. Uh, okay.
Yes, now it's much better.
Okay. Chromosomes unravel into filaments, and the nuclear membrane begins to appear. Cytokinesis occurs. That's all.
Okay, okay. Perfect. Um, okay, so chromosomes are decondensated now in this phase, and the new nuclear membrane is created. And the cell division is on the equatorial plane. This is what we already are speaking about, cytokinesis. Uh, Sometimes, or some resources, as the video showed us, cytokinesis is differentiated as an individual or separated phase. Some resources, they just involved cytokinesis into telophase. Okay, so it depends also on the resource you are studying from. Um, Basically, in this phase, or after the telophase, we have two identical daughter cells which are identical to each other, and also they are identical to the mother cell. Okay, so when the uh, so not it's a mother cell. Basically, the one mother cell divides herself into two identical daughter cells. So that's basically the outcome. Okay, I don't want to confuse you. It's similar to some third cell. Prophase, metaphase, anaphase, and telophase. That's basically what's going on during mitosis. Okay, here on the next picture, we are able to see how does it look under the microscope. Okay, so the phases, what do you think? How do what are the phases on the picture named? What do you think? The first one. What is this? Which phase?
Prophase.
Prophase, exactly, because there is a condensation of chromosomes, and this is the like drawing, and this is a real life prophase. The next one, what are your thoughts?
Metaphase.
Great, it's metaphase, exactly. And why do you see how the chromosomes are aligned in the equatorial lane, so basically in the middle of the cell, and there are some lines or fibers, those are spindle fibers, which are attaching the chromosomes to their centromeres. Okay, next one.
Anaphase.
Great. Anaphase. On this picture, we see how they are turned, pull apart to different poles. And this is basically exactly the same thing what we are able to see on this picture, how the chromatids are pulled apart to the different poles. And on the last one, the cell division is already finished. Okay, so it's already after the telophase and after cytokinesis. This is the picture. What do you think? Is this the picture of animal cell or plant cell? If you check the last one.
I think plant.
Plant. Why do you think plant, Sora?
Because the movies say that plant cells make wall,
Yes.
middle.
Yes, they make a cell wall in between them, and we are able to see the cell wall and the structure, how it is squared. This is not the typical shape of, or this is not the typical to have animal cells after division look like this. Okay, so exactly, this is a plant cell and this is a cell wall of plants between them, because they started to create in the late phase or in the cytokinesis of plant cells, we create or they create a cell wall between them from the center of the previous cell. Perfect. Okay. We still have some time. But we are, I think, more or less done with mitosis. However, for meiosis, it won't be enough time to cover it in 6 minutes. Um, okay, guys, let me check what we can do now, because I think it would be useless to start with meiosis now, because it will take more time. However, I can ask, would you like to see a short video now about meiosis, about as an introduction to what we are going to do on Monday, or no? Who is yes, no? Okay, so most of you went for yes, so we will see a short video regarding meiosis, at least to have an introduction to the topic we need to cover, and then we will be done with the session for today.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase 1 starts with a diploid cell. Its chromatin contains two uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of four sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with four genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has two stages of cell division called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce two haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces four genetically different haploid gametes. And here are some key points to remember about prophase 1. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Okay. Okay, guys. So that was it. Yes, and we can end session now. Please, guys, I have just one. Sorry, I was muted. So yes, we are done with the session. I have just one question. Is it helpful if we, for some specific topics which are very abstract, like mitosis, replication, translation, transcription, and so on, uh, we use videos immediately in the session, and then we also discuss the processes together? How are your feelings regarding that? Is it okay, or would you like to not do them? Uh, Okay, so it's helpful for you, I see. Okay, guys, just one thing. We will continue with the meiosis and next week. And when we are done with the next week, we will continue with the genetics of biology of genetic in the biology. And yes, so most of you went for genetics when I asked yesterday. Also the other group. So we will continue with genetics, and I will fix the problem regarding the recordings on YouTube during the weekend, okay? In order you will be able to see the recordings already, hopefully already next week. So I will try my best. Do you have any questions before weekend? No. Okay, guys. So there will be again no homework during weekend. I will upload just the videos we saw today, and if you want to see them again, you can. Please just enjoy your weekend, rest, and gain some energy for following weeks. Okay? Thank you very much, and see you soon.
Thank you.
You're welcome. Bye.